Thank you very much for coming to my talk. My name is Elzbieta Gralinska. I'm working at the Max Planck Institute for Molecular Genetics in Berlin. And today I would like to present you our work on association plots, which is a method for visualization of cluster-specific genes from high-dimensional single-cell transcriptomics data. When working with single-cell data, we quite often have a question which genes are overexpressed in a cluster of cells. And we can call such genes cluster-specific genes or marker genes. And these are the genes with unusually high expression in a cluster of cells in contrast to other clusters. And even though there exist methods for identification of such genes, usually the problem is that usually as an output, they provide a very long list of genes, and then we have to apply some thresholds, and this is how we can come up with the list of interesting genes. Of course, visualization of such results or gene cluster associations would be more helpful to get more insights into the data. But the problem with single-cell data is the fact that such data is usually very high-dimensional, so we have information from thousands or even millions of cells. And that's why we usually have to apply some dimensionality reduction methods. And by doing this, we reduce the amount of information from our data. And that's why we think that it would be useful to have a new approach for visualization of gene cluster association information also from higher dimensions. That's why we have addressed this problem, and we developed a method which we call association plots. Here on the right side, you can see an example association plot, which is generated just for one cluster from our input data. Each circle in this association plot represents one gene, and the genes are colored according to the strength of the association to a selected cluster of cells. So the genes of interest, or the most cluster-specific genes, would be located in the right bottom part of the association plot. So this would be these genes. And the most important thing about association plots is the fact that we are able to represent the data from higher dimensions in this planar plot. The question is, how can we achieve this? Namely, to generate association plots, we use a method which is called correspondence analysis. Probably some of you have heard about correspondence analysis, but this method is not very common in biological sciences, and that's why we decided to use correspondence analysis, which is in fact, it's a method similar to PCA. However, there are some differences. Namely, we are able, by applying correspondence analysis to our data, we are able to represent both the genes and the cells in one space. And the location of the genes and the cells tells us already something about the associations. Namely, in correspondence analysis space, genes which are located in the direction of given cluster of cells are overexpressed in this cluster of cells. And that's why we can use this for looking for cluster-specific genes. Namely, if we would be interested in finding genes which are specific for this orange cluster of cells, we would have to find the centroid of this cluster, and then we can draw a vector from the origin of the space towards this centroid of the cluster. And then what we do is we project the genes onto this vector. And then what we do is we measure the distances between the origin and the cluster centroid, and then we also measure the length of the projection of the gene onto this vector. And then by doing this, for all of the genes, we obtain an association plot in which each gene has two coordinates. And now it is obvious already that the location of the gene in the association plot tells us already something about the strength of the association of a given gene to the cluster of interest. And we can also go one step further and develop a method of measuring the strength of this association. Namely, we have invented something what we call S-alpha score, which helps us to estimate how strong is the association between a given gene from the association plot with a given cluster of interest. And to give you an impression of how does it look like, I highlighted 5 example genes with different S-alpha scores. The one on the right side, this is the gene with the S-alpha score above 2. For this gene, we can see a strong overexpression signal in the B cell cluster. And this is the cell cluster which I use for generating the association plot. And then the smaller the S-alpha score gets, the smaller is the overexpression signal in the B cells. And on the left side, we can see a gene which doesn't have any S-alpha score, and we can see that this gene is strongly overexpressed in other clusters other than the B cells. We can use S-alpha scores also to find novel marker genes in our data. This is again the same association plot which I showed you on the previous slide. In this case, with the gray color, I highlighted the genes which are known to be B-cell enriched. These are the genes obtained from the human blood atlas. And if we would be looking for novel marker genes characterizing B cells, we would have to look for genes with high S-alpha score, but these genes shouldn't be annotated as B cell markers. And for example, to give you an example how this look like, here I highlighted one gene with the highest S-alpha score. This is link RNA. This gene hasn't been annotated yet as B cell enriched. And we can see a strong overexpression signal. This gene hasn't been well characterized yet. It's mainly described in the context of cancer studies or cancer cells. And to my knowledge, there is only one source or one study which claims that this gene is B-cell marker. Of course, this is an illustration of how to use association plots for finding cluster-specific genes or novel marker genes, because if you would like to find proper novel marker genes, then we would have to spend more time on the predefined list of marker genes. We can also use association plots to annotate the cell clusters from our data to known cell identities. For this, for all of the cell clusters from our data, we can generate association plots. Then we can also, in the association plots, we can highlight the marker genes for cell identities or cell types of interest, and then the marker genes which will be located in the right bottom part of the association plots. These would be the marker genes for a given cell identity. And this is how we can use association plots for annotating the cell clusters to cell identities. With this, I'm at the end of my talk. Today I presented you association plots, which is a method for visualization of gene cluster associations. We can use association plots for revealing cluster-specific genes from high-dimensional single-cell data. We also introduced the S-alpha score, which is a measure of the strength of the association of a given gene to cell cluster. And also what I haven't mentioned today is the fact that we developed a bioconductor package, which you can easily use for interactively investigate the data and to generate the association plots and finding the gene markers in it. With this, I'm at the end of my talk. I would like to thank, first of all, Martin Winkron, who was my supervisor during my PhD, and with whom we developed association plots together. I would also like to thank Clemens Ko and Vida Sukhanan-Fadakar, who helped me a lot with the bioconductor package and with the Shiny app, which implement the concept of association plots. And also, thank you very much for your attention, and I'm happy to take questions.
Hi, I had a question about the inputs to your APL or package. Is it like a Seurat object, or is it, what is it?
Yes, we can also use, so first of all, we can use just the input matrix in the sense that this is the table of cells and the genes. We can also use the Seurat object or a single cell object.
Thanks.
Yep.
Hi, thank you for wonderful talks. I have a simple question. Maybe I missed some part of your explanation. So you developed the association plot. Maybe that can well represent each cluster because you calculate the association of the center of the cluster. But how can you define that genes as specific for that cluster? So I think I missed some part of the. Is there some method to compare each cluster associated plot or something to define that gene as specific for that cluster?
Can you repeat the last sentence, please? I haven't.
Yeah. So the main purpose of your work is to define a specific genes for each cluster, but the association plot picking a set of genes which can well explain or well represent some for each cluster. So how can you tell that gene is specific for that cluster? Is there a comparison process to tell that gene is specific or mutually expressed in each cluster?
So in the association plots, what I didn't really talk about is the fact that so when we're in the correspondence analysis space, then we identify the cell cluster of interest, and then what we do is in fact we look in the direction of this cluster, and then we project all of the genes onto this vector connecting the origin and the centroid of the cluster.
Yeah.
And then we know, like based on the theory of correspondence analysis, we know that all of the genes which will be specific for this selected cluster of cells will be located, first of all, like very close to this cell cluster, and also the angle between the gene and this vector will be very small. But we also, what I haven't mentioned in my talk is the fact that we also can make the conclusions about the expression of a given gene also in all other clusters, even though we generate an association plot only for this cluster of interest. And for example, here in this association plot, you can see the genes which are located on the left side, and they have a very high Y coordinate. And I can already say, based on this association plot, that these are the genes which are specific for some selected cluster of cells. And the genes, for example, which are not overexpressed in any of the clusters and which are uniformly expressed across all of the cells, these genes will be located here, so very close to the origin.
Oh, but that is the observed within one cluster.
Yes.
So each association plot is generated only for one cluster, but it also contains information on the expression in other clusters. So this is this whole magic behind association plots.
Okay.
And also, so what we recommend is to play with the association plots using our package, because then one can really explore how the location of the genes in the association plot, what it tells us about the expression about in the other clusters. So for example, for me, like I know already like the location of the gene tells me exactly where this gene might be also overexpressed, or what is its expression pattern across all of the cells. But to get more intuition about this, it's just worth to play with the association plots.